Hello students, hope all of you are fine. So dear children, we are doing respiration in plants and today we are going to continue the oxidative phosphorylation. Okay, we are doing electron transport system, or we can say electron transport chain. In that we had discussed how the exchange of ions and the you can say the hydrogen ions, okay, and how the oxidation is going on, how the ATP synthesis take place. Okay, so now we will discuss the oxidative phosphorylation in case of electron transport system. Now, oxidative phosphorylation is the synthesis of energy-rich ATP molecules with the help of energy liberated during oxidation of reduced coenzyme, that is NADH and FADH2, that are produced in the respiration. The enzyme required for the synthesis is called ATP synthase. Okay. Uh, it is considered to be the fifth complex of the electron transport chain. Now, ATP synthetase is located in F1 or head piece of the F0 and F1 or elementary particle. Okay, so you can see here we have the ATP synthesizing F0 F1 particle. Okay, so the particles are present in the inner mitochondrial membrane. ATP synthetase become active in ATP for formation only where there is a proton gradient having higher concentration of hydrogen ion or protons on the F0 side as compared to the F1 side. Now this is chemoosmotic hypothesis of Peter Michel, okay, which was given in 1961, and he won the Nobel Prize in 1978. Increased proton concentration is produced in the outer chamber or the outer surface of the inner mitochondrial membrane. Now, this is done by pushing of the proton with the help of energy liberated by the passage of electron from one carrier to another. Transport of the electron from NADH over the ETC, okay, that is electron transport chain, help in pushing the three pair of protons, that is 5 pair as per last estimate, to the outer chamber, while two late, okay, that is through the latest estimate, okay, that is 3 pair of protein are sent outwardly during the electron flow from FADH2 as the later donates its electron further down to the electron transport chain. Higher proton concentration in the outer chamber cause the proton to pass inwardly into the matrix or inner chamber through the inner membrane. The latter passes, okay, possess special rotating proton channel in the region of F0 of the F0 F1 particle. The flow of proton through the F0 channel induces F1 particle to function as ATP synthetase. The energy of the proton gradient is used in attaching a phosphate radical to ADP by high energy bond. This produces ATP. Oxidation of one molecule of NADH2 produces 3 ATP molecules, while a similar oxidation of FADH2 forms 2 ATP molecule. Now we will discuss the balance sheet of the ATP. Okay. Now, balance sheet, what is balance sheet? There is net gain of 2 ATP molecule during the glycolysis and 2 ATP, that is we can say GTP, okay, molecule during the double Krebs cycle. Glycolysis also forms 2 NADH2. Now it is reducing power, okay, its reducing power is transferred to mitochondria for ATP synthesis. For this, a shuttle system operates at the inner mitochondrial membrane. Okay, NADH2 is transferred to NAD, then NADH2, okay, and NADH2 is converted to FAD, then FADH2. The former operates in liver, heart and kidney cells, that is NADH2 NAD, then NADH2. This transformation take place in liver, heart and kidney. No energy is spent. The second method, that is the NADH2 FAD and FADH2, this formation occur in the muscle and the nerve cells. Okay, it lowers the energy level of 2 NADH2, 2 ATP molecules. A total of 10 NADH and 2 FADH2 molecules are formed in aerobic respiration. They help in formation of 34 ATP molecules. The net gain from the complete oxidation of molecule in the formation of 34 ATP molecule from the glucose in muscles and nerve cell is 36 molecule, that is 10 NADH2, okay, is equal to 30 ATP. 2 FADH is 4 ATP, and 4 formed by the substrate level phosphorylation of glycolysis and Krebs cycle, and 2 consumed in the transport of the NADH2 molecules into the mitochondria. In aerobic prokaryotes, okay, heart, liver and kidney, the 38 ATP molecules are produced per glucose molecule oxidized. Passage of ATP molecule from inside of the mitochondria to cytoplasm is through facilitated diffusion. Since one ATP molecule store 34 kilojoule or 8.15 kilocalorie per mole, we can say it is 7.6 kilocalorie or 3.8 kilojoule according to Browse. Okay, the total energy trapped per gram molecule of the glucose is 1292 kilojoule or 309.7 kilocalorie with an efficiency of 45%. Okay, the rest of the energy is lost as heat. Okay, now let's discuss the significance of the Krebs cycle. Okay, so Krebs cycle is the major pathway for the synthesis of reduced coenzyme and control release of energy during respiration. It is common pathway for oxidative breakdown of carbohydrates, fatty acids and amino acids. Amino acid enter the Krebs cycle directly as glutamate, that is for alpha ketoglutarate and aspartate for oxaloacetate after their deamination. Okay, now fat produce fatty acid and glycerol. Glycerol is phosphorylated and oxidized to form coenzyme A and to enter Krebs cycle. Acetyl coenzyme A provide 2 carbon compounds for the synthesis of fatty acid, cutin, aromatic compounds and isoprenoids for the formation of phytol chain of the chlorophyll. Then carotenoid, steroid, terpenes, okay, gibberellin, etc. Alpha ketoglutarate of Krebs cycle produce an important amino acid called glutamate. Succinyl coenzyme A take part in the synthesis of pyrrole compound of chlorophyll. Cytochrome and phytochrome oxaloacetate produce another important amino acid called aspartate. It also forms pyridine and acetate, okay, pyridine and alkaloids. It forms GTP, which is an important component of signal transduction, okay, system. Now Krebs cycle is amphibolic, that is catabolic as well as anabolic because it produces number of intermediates for anabolic pathway. Okay, so with this we conclude the Krebs significance of the Krebs cycle. Okay, in other video we'll discuss about the amphibolic pathway of the Krebs cycle.